NeuroCave Plus, web-based 3D visualization of biological networks. Abstract. NeuroCave Plus is an interactive web-based tool for visualizing biological networks in 3D space. It is designed to support a range of bioapplications, including brain connectomies, cell-type relationships, gene co-expression networks, and protein interaction networks. It provides an immersive, interactive visualization experience for exploring large-scale datasets, a versatile visualization platform that allows for easy adjustments in glyph scaling and simultaneous comparison of datasets with varying densities and scales. It enables users to efficiently search and highlight nodes, compare various datasets using color coding options, and utilize animation controls to emphasize important regions and connections within the visualization. With these features, NeuroCave Plus provides a powerful and versatile platform for exploring complex biological networks in 3D space. Introduction. The complexity of biological systems has led to the development of advanced visualization tools that can aid researchers in exploring and understanding these intricate networks. NeuroCave, a well-established immersive web-based connectome exploration platform, has been instrumental in analyzing network properties in the context of neuroscience. However, there is an increasing demand for a more versatile platform that can handle a broader range of biological networks, such as gene expression patterns in different brain regions, neuronal circuits, cell types, and tissue samples, as well as discovering relationships between cell types based on network theory analysis of gene expression or protein interaction networks. In this paper, we introduced NeuroCave Plus, an enhanced version of NeuroCave specifically designed to address these requirements. NeuroCave Plus extends the capabilities of NeuroCave to cater to a variety of biological networks, making it a powerful and versatile tool for researchers in diverse fields of biology and neuroscience. By providing an integrated platform for visualizing and comparing gene expression patterns and protein interactions, NeuroCave Plus enables researchers to identify key relationships and network properties that can offer insights into complex biological systems. We present the novel features of NeuroCave Plus, including its enhanced glyph styles, side-by-side comparison tools, and support for various types of biological networks, demonstrating how it can be an indispensable resource for researchers working on tasks such as comparing gene expression patterns in different brain regions, neuronal circuits, or cell types, and discovering relationships between cell types, or tissue samples based on network theory-based analysis of gene expression or protein interaction networks. Related works. Several immersive tools are available for visualizing biological networks in 3D, such as VRNetzer, CellExel VR, Cytoscape and StarMap. VRNetzer has a VR module built on Unreal 4 and a general flat GUI built on JS and D3, with a backend MySQL/Python analytic module. However, its VR capabilities are separate from the web UI, limiting the integration of both visualization approaches. CellExel VR, on the other hand, is written in Unity and Steam VR, and it includes VR functionality. It is specifically designed for single-cell transcriptomics and supports the comparison of two or more networks in the same 3D virtual space. However, CellExel VR does not have a web interface like NeuroCave Plus and lacks a flat side-by-side display for comparing networks. StarMap is another WebGL app that visualizes large numbers of spheres representing biological data points but lacks selection or connection features, and its VR capabilities are limited to a smartphone in a Google Cardboard enclosure. This allows for depth perception and head tracking but not hand tracking or the use of modern controllers such as Oculus Touch, offering only arrow keys and SID keyboard navigation. The popular biological network visualization tool Cytoscape has rich community support, and its modular architecture and App Store model have made it the industry standard among open-source tools of its class. There is a 3D visualization module called Cy3D, but it lacks the flexibility and ease of use required to provide a good user experience with large graphs or in comparison studies. NeuroCave Plus stands out as a fully web-based immersive tool, written in JS with the Node.js framework and the D3 and 3G graphics libraries. It provides a pair of side-by-side 3D network visualization viewports, each with an integrated WebXR VR option at the click of an Enter VR button, suitable for integration into web-based data processing environments such as Galaxy. This integration allows users to visualize and compare different networks or different representations of the same network simultaneously, offering a lightweight and versatile solution for immersive exploration of a wide range of biological data. Visual design. NeuroCave Plus was primarily designed to give the user the flexibility to use the linked side-by-side view, not only as a comparison tool but to provide two different general-purpose views of the same data. This allows for the use of one network as a reference for the context of a related network. For instance, one dataset, such as a gene co-expression network, could be represented with the cube glyphs, and another one, such as a brain connectome or cell lineage map, could be represented with the spherical glyphs. This way, connections within or across networks can be explored, with across-network connections possibly representing gene sets expressed by cells in a single-cell transcriptomic study or in a sample of brain tissue. Conversely, within-network connections would be interpreted as gene co-expressions in the gene network, or in the cell network as direct cell differentiation subtypes, or in a brain connectome, of axon bundles transmitting signals between two brain regions. Additional generalization features allow analysis tasks beyond comparison of two networks. Where NeuroCave had a single slider controlling the glyph sizes, NeuroCave Plus has two sets of independent glyph size control sliders for left and right view windows, allowing users to control sizes of cube and sphere glyphs for the left and right view windows. This is useful for neuroscientists comparing brain connectivity between or focusing on a specific hemisphere. This feature also allows general users to view two different datasets simultaneously in each of the windows, compensating for point density and scale variations on the fly. Intragroup and intergroup connectivity, edge display toggle, and threshold adjustment for examining connectivity patterns between cells, samples, brain regions, and genes in genomic datasets. Separate control of ipsilateral and contralateral connectivity. Edge display gives users more control over the edge visibility thresholds, allowing them to focus on the most significant connections or get a general sense of the connectivity patterns. Decoupling the color code selectors in the two windows allows for various use cases, including independent control of region color visibility and highlighting specific brain regions in their connectivity. Different color codings of the same dataset, with dual-faceted views allowing selection of specific regions and their connections in each window to explore the intersections between the two color codings. Displaying different colorings of genomic datasets to draw correlations between metrics such as gene module membership and gene centrality. Displaying complementary datasets in each window for intermodal exploration of networks, with customized color codings for each dataset. The search bar can be used to locate specific brain regions or genes of interest and highlight their connections or correlated regions in the visualization. The 3Gs library allows for precision HSL color control, which is used to draw attention to selected or otherwise important glyphs while preserving the color coding by oscillating the luminosity and saturation while mostly leaving the hue channel alone. Animation amplitude and frequency control for pulsing and flashing of selected and connected nodes provides a means to highlight important brain regions and their connections, allowing for edge lines to be dimmed or completely removed to reduce crossing edge clutter. There is an edge opacity control slider and a pair for animation amplitude and frequency, providing the user with a fully customizable network connectivity display. The original NeuroCave 3D platonic solid-based representation of clustering data is also useful for exploring the hierarchical clustering of cell relationships or gene expression patterns, as well as neuronal circuits. Here are a few more specific use cases for NeuroCave Plus. Exploration of the connectivity pattern of brain regions and genes in 3D space, with the ability to highlight specially annotated gene types such as gene fusions, Leukrina, Serkrina, and protein coding mRNA expression patterns in the brain, alongside the brain regions of interest in which they are expressed. Visualization of large-scale spatial or single-cell omics studies, with independent glyph size and color coding control to adjust for differences in point density and scale when comparing and contrasting single-cell or microdissection data across different regions of the 3D embedding. Comparison between clustering algorithms such as Louvain, Place, and Vicina on the same dataset, exploring the differences in the clustering results in the gene modules identified by each algorithm. Exploration of the connectivity patterns of genes in 3D space, using different connectivity matrices, derived from different types of data, such as gene expression, protein-protein interaction, or KEGG pathway data. Visualization of two protein interaction networks, perhaps a healthy and diseased pair, imported into one shared immersive interaction space, with one be represented with spheres and the other with cubes, so that when they were superimposed, it would be clear where the differences lie. To still be able to perform comparison tasks while using the two viewing windows in this way, some features were added to support the representation of two distinct datasets in the same immersive space. Future work. Performance optimization. Level of detail. The software should implement a level of detail, LOD, approach, rendering particles until they are close enough to resolve into spheres, cubes, or triangles. Sparse matrix implementation. The software should utilize a sparse, rather than dense, matrix for handling large connection matrices, reducing memory usage and improving performance. Other future developments include enabling edge bundling, implementing more advanced coloring schemes, and extending the software to visualize larger datasets. Encapsulation of NeuroCave Plus into an interactive visualization module for Galaxy will streamline the data processing and import process, allowing users to easily design their own workflows and seamlessly visualize their results as a 3D space embedded with a VR option. Integration with large language models could help identify more complicated network structures simply by typing natural language into the text box. Conclusion. The advanced 3D visualization software presented in this paper offers a powerful tool for researchers investigating complex biological systems, such as brain connectomies and gene networks, providing users with a high level of customization and control, facilitating in-depth analysis and exploration of anatomical and genomic data. It provides a comprehensive and interactive platform to explore and analyze complex neurogenomic datasets by visualizing brain regions and genes in 3D space and allowing for easy comparison and exploration of different datasets, with various dimensionality reduction embeddings and color codings. The decoupled control of glyph size, color coding, and edge display controls provide users with the ability to customize the visualization to their specific research needs, while the search bar and animation controls provide a means of highlighting specific genes or brain regions of interest. NeuroCave Plus, web-based 3D visualization of biological networks. Abstract. NeuroCave Plus is an interactive web-based tool for visualizing biological networks in 3D space. It is designed to support a range of bioapplications, including brain connectomies, cell-type relationships, gene co-expression networks, and protein interaction networks.